In this video, we're going to look at an overview of genomic DNA extractions. So essentially, first, I want you to think of a cell and all the components within it, and then imagine you're going to try and purify or isolate one aspect away from all the rest, because that's essentially what we're going to do in this session. So consider an animal or a plant cell that you've seen before. Look where the nucleus is in each case. Nucleus is where the genomic DNA is housed, and that's what we need to isolate away from every other aspect and component of the cell. If we think of DNA, we already know it's hereditary material. We know it lives in the nucleus, the genomic aspect. We know there's actually a small amount in your mitochondria as well. It's a double helix with a sugar phosphate backbone, and that phosphate gives the DNA a negative charge, something we're going to meet in a future practical session. It's composed of certain bases, and A complements with T and G complements with C. We know there's approximately 3 billion bases in human cells, and the order or the sequence of those bases is the determining factor. So we replicate our DNA in preparation for mitosis. So we already know a lot about the information. What we're going to do today is try and get a kiwi and isolate genomic DNA from some kiwi cells. So firstly, you're going to get a kiwi, peel it, and slice it into small pieces. So imagine you have your kiwi cell and you're trying to isolate this genomic information. Well, we're going to add some extraction buffer or lysis solution, and we're going to add some physical disruption to break down the membranes. And because that will happen, we're essentially releasing the cellular components. We're then going to filter the cellular mixture through cheesecloth or muslin, and that way we're able to gather a filtrate, a liquid filtrate of our kiwi solution that's been filtered. From here, you're required to use 6 milliliters. So you're going to transfer 6 milliliters into a new tube of this filtered kiwi solution. And to that solution, you're going to gently layer 4 milliliters, so pipette down the side of the tube, ice cold 95% ethanol on top of that filtered kiwi solution. So pipette it nice and gently into the tube down the side. And then you're going to observe and record and document your findings. So keep an eye once you've added the solution, where the both solutions are kind of meeting. I want you to keep an eye on that over the next few minutes once you've added that ice cold 95% ethanol and see what happens. Okay? You should begin to see your DNA precipitate at that point. Your instructor will be able to guide you exactly on what you see and what you're actually visualizing at that time. So just to recap, you'll peel and slice your kiwi. We're going to add the extraction solution and some physical pressure to disrupt it. We'll filter it, remove 6 milliliters of the filtered solution before adding 4 milliliters of ice cold 95% ethanol. We'll observe, document, record what we see, and we're going to spool our DNA into a fresh tube. So before you go, when you've transferred it into an Eppendorf, please label that accordingly and keep adding water until your DNA dissolves, recording how much you've added. And please give your tube or submit your tube to your supervisor before you leave, because you need this sample in future weeks. And then clean your bench, dispose of any materials and consumables accordingly.